Well, I officially welcome all of you guys in this series of neuroanatomy. Well, the first lecture was very basic lecture about the terms that will come across in this whole series. So now we are going to dive into the neuroanatomy from very basics. So what I want you to take in from this lecture, actually after this lecture you should be able to describe the basic classes of cells found in central nervous system, and you must be able to characterize the functional microanatomy of neurons. Actually you must be able to differentiate the different parts of neuron and the function of neurons. So I call it a functional microanatomy. And you must be able to describe the microanatomical composition of gray matter and white matter, which we already talked about in the last lecture. Actually, you know, the neurons are cells, and well, cells group together, they make tissue. And when neurons group together, they make neuronal tissue. Whenever we talk about neuronal tissue, we actually mean about gray matter. When we say gray matter, we actually mean neuronal tissue, or we say white matter, we are talking about neuronal tissue. So I'll give you this concept also in the end of this lecture. So now we'll be talking about neurons in this lecture. Neurons are actually basically primary processors of neuronal signals. And neuroglial cells, if we talk about neuroglial cells are actually basically supporting cells of neurons, or that support the electrical and chemical function of neurons. So we'll talk about neuroglial cell in next lecture. In this lecture we will only talk about neurons. And if we talk about brain is very rich in the blood supply. It is a highly vascular organ because it needs the blood supply and it needs the nutrients. It needs oxygen and glucose and elimination of the carbon dioxide. So for the supply of blood, there will be vascular endothelium also. Then when you see brain tissues, you will see vascular endothelium, right? So let's look into neuron. So let me make a very fundamental remark, or a very basic remark, that neuron is a fundamental unit of function in CNS. That's why we'll talk a lot of time. That's why we'll give a lot of time to neuron, talking about neuron. If we talk about neuron, as neuron is a cell, so it has all the metabolic machinery that is common to other somatic cells. So in this aspect, a neuron is like all somatic cells in this aspect. But if we talk about neuron, it is a bit different from other cells in some other aspects. If we talk about its diversity in its form, in its morphology. Whenever we talk about form, we also think of physiology, because form and function are equally related. So it is also actually different in form and function also. So bioelectrical properties of neuron are also different from other cells. And if we talk about a specialization for intracellular communication, there you will see synapses between neurons. And whenever we talk about synapses, we talk about junctions or meeting point between neurons. And you will see that neurons are specialized for the formation of neurotransmitters. So we'll also talk about it in this lecture. So let's actually move on to take survey of neuron. Let's do a survey of neuron. Let's start from cell body. So as all cells, neuron has a cell body. You can clearly see the cell body here. The nucleus is clearly visible. As all cells have cell organelles, neuron also has cell organelles. But neuron is very rich in certain cell organelles. For example, endoplasmic reticulum, neuron is rich in ER. For example, both type of ER, smooth and rough endoplasmic reticulum, because it needs to synthesize protein and lipid to maintain the synthetic function of neuron and to maintain the morphological function, morphology of neuron. So it has the cell body, it has the endoplasmic reticulum. On the same side, it also has a mitochondria to synthesize ATPs. So it has all the structures that a cell has, that a typical cell has. So if we see the cell body of neuron, you see a lot of projections coming out from the cell body of neurons. These are actually protoplasmic projections from the cell body of neurons. If we talk about them, if we talk about projections which are small in size, which are 50 micron or less than 50 micron, we call them dendrites. So we are going to talk about them. Actually, dendrites are the input, or the places where neuron actually connect, or input zone, the places where neuron receive information. So whenever you think of dendrite, you may think of input zone, you may think of a connection between neurons. So actually it is a connection between neurons, and dendrites are actually making the surface area of neuron wide. they are actually increasing the surface area of neuron. If we look at the microanatomy of dendrites, if you look at microanatomy of dendrites, you will see that dendrites have spines. For example, imagine it is dendrite. Forgive me for my bad drawing. Imagine it is your dendrite, and there will be like this spikes. It may be mushroom shaped, or it may be like straight like this. So actually, these increase the surface area of dendrite of even a single neuron. You know, there are some dendrites which are smooth in their appearance. They are smooth in their appearance. Actually, those dendrites are of those neurons which makes connection with those cells that inhabit the action. These dendrites are actually going to inhabit the action of those neurons in which they are connected to. So actually, this is this is all about dendrites. So let's talk about another process, another cytoplasmic process that arises from neuron, which we call it axon. Axon is, you know, very special kind of cytoplasmic process. It is actually kind of process which transmits or which conducts the information from the cell body. Actually, it conducts the information in form of electrical signals, which we call it actually action potential. So like this information will be conducted in axon. So axons are actually different in sizes. Actually, axons may be of 100 microns, but it can be of very large size. For example, the axon in your cerebral cortex may be so wide that it may be connected to your spinal cord. So it can be even 1 meter long. So axon vary in size, but we talked about dendrites. Dendrites are small in size. So axon actually conduct electrical impulse, so we call it a conducting zone. So the tissue where we find axon, we call it a conducting zone, and where we find dendrite and dendrites, we call it an input zone. So let's talk about the synapses or axon terminals. If we talk about synapses or axon terminals, these are places where axons actually meet, where neurons meet. These are actually connection point between neurons. There actually synapse means junction or connection point between neurons. Actually, there are two types of synapses. There are two varieties of synapses, which we'll talk it about later on, but I want to give you just overview. Actually, there are electrical synapses and chemical synapses. If we talk about electrical synapses, in electrical synapses there are actually junctions between cells or junctions between neurons. Basically, typically there are gap junctions in which the electrical impulse or electrical signal is transmitted from one neuron to other neuron in the form of a charged particle or charged molecule. On the other hand, on the contrary, there are also chemical synapses which are common in the nervous system. If we talk about chemical synapses, you will see that there is one nerve ending, there is one neuron, there is other neuron, and there is a free space. We call this free space as a synaptic cleft or a cleft. So electrical impulse will come here from axon to this nerve ending, and from this nerve ending it will reach a neurotransmitter or some sort of substance that we'll call it neurotransmitter, and this neurotransmitter is a chemical. This chemical will go and attach on the receptors on the other nerve ending or on the other neuron. These receptors will convert this chemical signal into electrical signal. So this sort of actually synapses are called as a chemical synapses. So there are basically two varieties of synapses, electrical synapses and chemical synapses. So now let's put together actually neurons. For example, this is one neuron, this is other neuron. We are parallelly putting together this neuron in a parallel fashion. So when we put together neuron are cells, they make a tissue. So we this portion of neuron, this portion of a neuron where you are watching the cell bodies and dendrites. I told you in the first lecture, the combination of cell bodies and dendrites, we call it white matter. So this tissue, this neuronal tissue will be called as the white matter. And I told you in the first lecture that whenever we see collection of axon, we call it white matter, and where we see a collection of cell bodies and dendrites, we call it gray matter. I'm really sorry, I missed it. It is called gray matter and it's called white matter. So let's move on. Let's move forward. So when these neurons are parallelly joined, or when these neurons combine together, they form a tissue, neuronal tissue. When we see this neuronal tissue, it has some sort of gray matter and some sort of white matter. Where we see cell bodies and proximal dendrites, we call it gray matter, and where we see axons